All right, folks, so far we were talking about the microscopic anatomy of muscles, or specifically skeletal muscle, right? We started off with the muscle fiber, and then we realized that the muscle fiber is made of bundles of long organelles called myofibrils, and those myofibrils are basically made of chains of these repeating sarcomeres, and the sarcomeres are made of myofilaments. And the two proteins we focused on was myosin and actin. Well, now we're going to get into more details because we're going to use the sliding filament theory to help us understand how do muscles get shorter. We know that if the muscle gets shortened, then it is also contracting. So those details, we have to understand the proteins themselves. So first, we talk about myosin. And myosin is a thicker filament that's going to be anchored to the center of a sarcomere. So we had our little sarcomere here, and then we have the middle of the sarcomere. Well, the myosin is going to be anchored to the middle of that sarcomere. I'm trying to keep the same colors as we see in these areas here. And remember we said that muscles can only pull, never push? Well, the myosin is only going to want to pull. What is it going to want to pull on? It's going to want to pull on a rope, for example. Okay, but before we can understand where it pulls, we have to really get more details about the anatomy of that myosin. So myosin, again, is going to be anchored. It's a thicker filament anchored to the center of the sarcomere, and it has these little heads on both ends. Okay, we got a head here, and we have heads here. Again, we do it up here, we do it down here, here, and here. And it's these many heads, and sometimes it's being called the many-headed medusa of this myosin that wants to pull on something. Okay, now what is it pulling on? It's pulling on rope. Now what is rope like? Actin. Actin is basically this rope-like protein that's anchored to the ends of the sarcomere. So we're going to have actin that's at the end of the sarcomere. Let's say this is our end here. And some of the actin is going to be already pulled or connected to the myosin heads here. All right? And we can see that kind of displayed here. We see 3 sarcomeres, but if we want to kind of have our boundaries here, we see our myosin, and we see some actin, a little bit of overlap with the actin overlapping the myosin heads. Not completely in the middle, but a little bit of overlap, right? So what's going to happen is the myosin wants to pull on that rope of actin. And as it pulls on that rope of actin during a contraction, the actin is going to end up moving towards the midline of the sarcomere. So the actin moves this way and this way until it gets to the middle of that sarcomere. Why? Because it's getting pulled on by those myosin heads. But that pulling is only going to happen during contraction. And guess what? These filaments, the actin and myosin, they stay the same length. The only thing that's going to shorten is the sarcomere overall. So the entire sarcomere unit will shorten, but the length of the actin and myosin will stay the same. Now, how do we understand that shortening versus, and how do we understand the actin and myosin staying the same length and that shortening? Well, we have some analogies to help with that. So we have this general analogy and also just using regular sarcomeres. Of course, my brain's going to want to just use regular sarcomeres, but I'll talk about this analogy just in case someone actually likes it. Okay? So instead of just saying that the myosin is many medusa heads, they're going to say that the myosin is going to be the head and the arms that we see here. So here's myosin, and all these guys are little myosin. Then we're going to focus on the ends of a sarcomere. So here's our boundaries for the end of a sarcomere. That means we're dealing with one sarcomere here, and one here. And we can also see those ends are going to get shorter when we see the sarcomeres are here a little bit shorter, here shorter, and here shorter as well. Okay? And those same 3 sarcomeres are the same 3 that we see in our sarcomere analogy, which again, we see the boundaries here, so that's 1. Boundaries here, that's 2. Boundaries here, that's going to be 3 sarcomeres. Okay? You can also just write lines to help you follow that. And what do we notice here? We notice that the myosin head and arms are holding, well, really their arms are holding the actin rope. So we see that actin rope. Right? Whereas down here, we see the same arms, but guess what? We see the arms being extended. They're pulling on that rope. So they're pulling on that rope, doing what? Causing more slack in the rope itself. So here we see some kind of slack. When it gets pulled on, that rope has a lot of slack. And the slack is going to equal overlap here. So when we're looking at a sarcomere, even on the one I drew here, you see how it has a little bit of overlap of this rope actin here? Here we also see a little bit of overlap of the actin. Doesn't go all the way to the middle, but there is some overlap there. And then when it contracts or we get more slack, we see a lot of overlap, way more overlap of the actin going across as it gets to the middle. That's what contraction is going to do. That's going to help us understand the pulling of the myosin heads. So the myosin is going to pull the actin, creating more slack, causing, again, the sarcomere to shorten or get smaller. We see how it was longer here. Now it kind of got smaller there. And that's when contraction happens. Now, when contraction is not happening, what's going to happen to that rope? It's going to go back to its original size and have that tiny bit of slack or overlap. And that's the whole idea behind this. So let's look at a sarcomere here. This is where it's going to be relaxed. This is where it's going to be a contraction. Let's focus on the size of the middle sarcomere here. Again, we have our boundaries of our sarcomere. We have our myosin. We see all the myosin heads. Some are already attached to the actin rope from the top and the bottom on both sides, right? And then we see our actin starting from the end of the sarcomere, going a little bit overlapping some of the myosin heads, right, towards the middle. When the myosin heads are going, when a contraction is triggered, when the myosin heads start pulling on the actin, you can see we get way more overlap with that actin as it goes towards the middle. And that sarcomere gets shorter. You see the line is longer here versus when it's contracted, it's shorter. Okay? And as you can see, the sarcomere itself gets shorter, but once the muscle relaxes again, the actin myosin goes back to its normal length. Nothing has changed with the actin and myosin, so they stay the same length. The sarcomere is the thing that gets shorter. Now the question becomes, well, how does all this happen? How does the myosin know when to start pulling on the actin? Let's go ahead and get into that next. So remember, we want to know how does myosin pull on actin? When does it know when it's going to pull on actin for a contraction, right? Well, let's think about it. We have our muscle fibers. We know that we have the sarcolemma that is surrounding all of our myofibrils and our T-tubules that are, again, kind of creating canal or tunnels surrounding those myofibrils. And then that sarcoplasmic reticulum is also there to help that electrochemical signal get down deep into the myofibrils, right? What did we also say about the sarcoplasmic reticulum? It's going to store and release calcium. It's going to release calcium when that signal hits it, and it's like, okay, it's time for a contraction. And it's going to store calcium until the next contraction is ready to go. Let's get into the details of how that little piece happens. How is that calcium going to cause contraction? Well, first, let's look at our contractile proteins. We know that the myosin is a thick filament. The actin is a thin filament. Again, you can say many-headed medusa, or you can say actin is thin. But we need to be able to regulate when that myosin is going to pull on the actin. And we have to use regulatory proteins for that. There's going to be two proteins that are help with the regulation of this, okay? Tropomyosin, troponin. Tropomyosin is going to be something that does not like myosin. So even though it's a part of the name, you might think, oh, tropomyosin, they go hand in hand. No, tropomyosin is literally this thread-like protein that wraps around actin. So you see all this green thread that's wrapping around the actin? The actin are like these little beads here. Let me show you one of these beads that has a little opening. Guess what that opening's for? That opening's for the myosin heads to attach. And when you have this thread covering those openings, myosin does not attach. So this is going to block the myosin binding site on the actin. So again, you can call it an opening where the myosin is going to attach, or you just say the myosin binding site on this actin. See, myosin binding site. And as long as that tropomyosin is there, there is not going to be any additional pulling on this actin, all this, shortening of the sarcomere. There's not going to be any contraction, okay? But there's another protein that we see, these little globs of blue. And this is called troponin. These globs of blue here. And the beauty of troponin is that they're the little globular proteins, and what they do is they sit around the actual tropomyosin. And they regulate tropomyosin. It's like, okay, you can block the actin heads right now, or the myosin binding sites. You can block those right now. But when I tell you to move, you're going to move. Now, how does it actually tell the tropomyosin to move? It's going to bind to good old calcium. So when that calcium ion comes in, it's going to bind to calcium and it's going to open those binding sites by moving tropomyosin out of the way. That's what happens. That's how we regulate whether or not the contraction is going to happen. And once the tropomyosin, the thread-like proteins here, are moved out the way, myosin can now bind. Myosin heads can now bind, for example. Bind here, for example. And what is it going to do? It's going to want to pull. Pull on what? Pull on the actin, moving it toward the midline of the sarcomere so a contraction can happen because the sarcomere is getting short. That's exactly what happens. So now we need to make sure we can understand. There's easy little memory things here about the regulatory proteins. Tropomyosin, because it has myosin in the name, says no to myosin. Tropomyosin says no to myosin. Troponin opens the binding site. So the fact you see OPON, you can also think of open. That could be one way to remember. Troponin opens the binding site. Tropomyosin says no to the myosin. My way of remembering is the fact that I see myosin in the name means that it does not like myosin. So it blocks it. Okay? There's other structural proteins that you see here. Yes, we have the middle of the sarcomere. We'll get the details of that. We'll get the names and what this little zigzag line means. And there's also these proteins here. And these proteins are huge. These are elastic filaments, or proteins, specifically called titin. And they're going to help the sarcomere retain its shape. So remember what we were saying, I'll go back to our analogy that when it's relaxed, we have this shape. When it contracts, it has this shape, and then it goes back to the relaxed shape after the contraction is complete. Remember when we said that? Well, it's going to be the elastin that helps the sarcomere retain its shape. And you can see it's everywhere. It's attaching to the myosin, it's attaching to the actin. And so even though the myosin is pulling the actin towards the middle, to the midline of the sarcomere, causing a contraction because it's getting shorter, the elastin is going to allow it to, again, go back to its original shape. And this titin is very large. These are huge proteins that are going to help with that. And guess what? Remember we talked about some properties of muscle. It's going to be elasticity. Well, elastic filament, elasticity, there's where it comes in. Now let's get more microscopic. Because we need to be able to see how sarcomeres look when this is actually happening. What does it look like when it's contracted? What does it look like when it's relaxed? Our goal is to try to get into those details, at least have it in our minds and some kind of that. So the research sarcomeres were named for how they look on a transmission electron microscope, TEM. Now, a transmission electron microscope is really cool because it's going to be able to show the fine details of cells. So as you can see here, I can actually see a sarcomere based on this. I see the boundary of the sarcomere here, over here as well. I see the midline of the sarcomere. I see a dark region, and I also see a light region. Let's get into those details of what they look like. Another version of this sarcomere is down here at the bottom, our good old little graphic that we've been using this whole time. So first, let's talk about the ends of the sarcomere. These little zigzag lines here, that's what they're called. The Z-disc. They're at the end of the sarcomeres, the Z-disc. And you need to make sure you're able to label this so you can understand the changes that happen in these sarcomeres. So the Z-disc. And the Z is at the end of the alphabet, so you can think of it that way. But also I see that this is zigzag, so I think Z-disc for zigzag. Then let's talk about the middle of the sarcomere, called the M line. Okay? So this is going to be our myomesin protein that anchors the myosin. You can see, excuse me, as you can see, it's anchoring the myosin, allowing the myosin to do its job. So again, that's going to be our M line. Now we should know that this is going to be our myosin. And this is also our actin. We've seen those before in the previous, okay? And we also know this is our titin or our elastic filament. Now let's talk about the areas that are light and the areas that are dark. So first, let's talk about this. We see our Z line here, and then we see this area that is very light. Then we have this whole area here, a different color, that's very dark. We got to be able to talk about what the light area is called and what the dark area is called, and then be able to verify it on here. Okay, this one is dark. And it's also light over here, but we'll just focus on one side. Well, the light side is called the I band. We call that the light band. Sometimes you can just underline the I and light to make sure you're talking about the I band because it is light. And this is area with just actin. Now, since actin is a very thin filament, it's very light on the transmission electron microscope. So you can see all this here is just actin. And of course, we know that because the actin is going to be anchored at the end of a sarcomere, which is at the Z disc. And it's just going to be, again, very light in that area. We can also see it here. This is the I band because all we have is actin. All we have is actin at the end of the sarcomere, and then we have all the actin here. That's the brightness that we see on the transmission electron microscope. Then we have the A band, which is going to be the dark band. Sometimes you can just underline the A and dark, and you will realize it's talking about the A band. And this is the area with both actin and myosin. So this whole thing is considered the A band. Why? Because it has myosin and it has actin. Does it also have myosin in here? Yes, but we're talking about the areas that is going to be actin and myosin, which show up dark in these areas here, okay, on the transmission electron microscope. But you also might notice, what about the space here? What about the space in between? Let me get another color. This space here and this space here. Also, you can see like a little space here and also a little bit of brightness there, space there. That is called the H zone. Okay? And it looks like an H. That's called the H zone. And this area just has myosin, as we can see. And it's called the H zone because that's going to be a little area of brightness where you're like, hey, this is the dark region, but I see a little bit of brightness. That's because pele means bright. Okay? So now let's talk about contraction really quickly. Because you're like, well, great, we know how to label the sarcomere, but what's going to happen to each of these little things, each of these little lines and bands and zones when we actually contract? Are they going to get shorter? Are they going to get longer? Are they going to stay the same? Well, let's figure this out. Let's talk about this A band here. Very, very dark. Now, the A band is really defined by the myosin in it because that's why it's really dark. These thick filaments, right? But it also has some actin in it as well. As you contract, the myosin is going to pull on the actin towards the midline. So it's going to pull towards the midline. And that's only going to make the A band darker. So this is not going to have no change when you have contraction happening. Let's talk about something else that won't have any change. Well, let's think about the M line. The M line also wouldn't have no change. Why? Because it's not even moving. It's in the middle anchoring thing. So nothing's going to happen to it. Is there anywhere else we can say, oh, it's not really going to have any change? Okay, let's continue then. What about the I band? Now, the I band is all actin, right? What's going to happen to that actin? The actin is going to move toward the midline. It's going to keep moving toward the midline. So because it's moving toward the midline, that means all the actin that's in the I band area is going to be moving to the middle. And so what's going to happen to the I band? It's going to get shorter. So this area here is going to get shorter because all of its actin is going to move towards the midline. Okay? What does that mean? You can also say the actin is going to be more overlap of the actin. So the I band is going to get shorter because the actin is now going to be actively moving to the middle of the sarcomere. All right. Now let's talk about the H zone since we're already over here. So here's our H zone. We have all this like little brightness that we see here. And I can erase it just so you can see one side of brightness here. We have a tiny, tiny bit of brightness that we see on the transmission electron microscope. Now, what's going to happen when our actin gets towards the midline? What's going to happen to that H zone? Well, if the actin is getting towards the midline here and getting towards the midline here, then there shouldn't be any additional brightness because the actin and myosin are working together now. There's going to be a lot of overlap of actin here. So that little bit of brightness is going to go away. Does it automatically go away? No. It just gets shorter and then it disappears. Why did it disappear? The actin got to the midline because of the contraction. Okay, so just keep that in mind. So we did the I band. We know that's going to get shorter because the actin is moving towards the middle. The A band is not going to have any change. All you're going to have is more actin in the middle, which is going to still show up dark. The H zone is going to get shorter and then it's going to disappear because the actin goes to the middle. The M line doesn't change. What about the Z disc? So the Z disc is the end of a sarcomere. What do we know about actin? Actin is anchored at the end of a sarcomere. So you can also say actin is anchored to the Z-disc, right? That's what we know. So we know it's going to anchor the actin. Well, the actin is moving towards the midline during the contraction. So what's going to happen to the Z-disc? It's going to move. But does it change its shape or its size? No, it just moves. And then the elastin is going to put it back into the original shape of that sarcomere. That's it. So I want you to kind of just keep that in mind as we go into some of our worksheet examples next to end this video off. All right, we're going to focus on number 10 here. It says, imagine whether the myosin binding sites on the actin. So again, if I have my little beads of actin, I have a little myosin binding site within it. So it's going to imagine whether the myosin binding sites on the actin will be more likely to be exposed or blocked in the following two scenarios. So it's going to be exposed or blocked. If it's exposed, what does that mean? Before I even read the rest of it, that means tropomyosin is not present. Right? That means that thread-like protein has moved away from the actual bead, allowing for the myosin binding site to be open. Right? But if it's blocked, that means the tropomyosin is actually across the binding site here. So let me draw some pictures on my head. So let's say this is our tropomyosin thread. If it is blocked, this is what that's going to look like. And if it's exposed, then the thread is not blocking it. So it's kind of around it. It can be under here, just for example. Okay? And they're giving us two scenarios. Then it says, likewise, predict whether you would expect overall myosin binding to increase or decrease in each scenario. So let's think about this. If it's exposed, we know we're going to have some type of increase. Okay? If it is blocked, we know we're going to have a decrease in the myosin binding. That means the myosin binding site is open or is it closed? Exposed is open, blocked is closed. Blocked also means that tropomyosin is going to be blocking that site. Open means tropomyosin is not blocking that site. Now you could go even further and say, well, what's stopping the tropomyosin from exposing it or blocking it? Well, think about it. We need troponin. And if it's exposed, that means, we need another color, but troponin is binding to calcium to expose the myosin binding. When it's blocked, that means the troponin is not exposed to calcium ions from the sarcoplasmic reticulum. Okay? So let's look at our two scenarios. There is no tropomyosin present on the actin filament. So if there's no tropomyosin present, that means we're talking about an exposed situation. So the myosin binding sites are going to be exposed. If they're exposed, will the binding increase or decrease? Increase. You see how, because I'm breaking down and adding notes to this question, I can automatically get the answer, right? Sometimes that's what it's going to take. I recommend, just like I said before, that you are going through and using your notes to break down pieces of this question, add details where you can, because you want to be able to understand it. The calcium binding sites on troponin molecules are non-functional. So it says the calcium binding sites on the troponin molecules are non-functional. So that globular protein troponin can't bind calcium. If it can't bind calcium, it can't expose the myosin binding sites. So that means it's going to be blocked. Binding is going to decrease in this scenario because, again, the myosin binding sites are blocked. Okay? The way they presented the information just in this question, the calcium binding sites on the troponin molecule, they're adding in and kind of mixing together all this terminology that we just learned. You want to make sure you can clear it out in your head for yourself to be able to approach that question. All right, folks, I'll see you in the next video. We'll talk about muscle contraction, basically getting more details about how muscles actually contract. See you there.